Hello everyone, Dr. Alex Vasquez here to provide you some more information on our upcoming International Conference on Human Nutrition and Functional Medicine, to be held here in beautiful Portland, Oregon, September 25th through the 29th. In one of the last videos, I provided you some updates about the venue, which of course is here in Portland, Oregon. Here's our convention center, and as you can see in the lower right-hand corner, that's the light rail system that will take you directly from the airport to the convention center and the nearby hotels where we have discount blocks available. Right next to the convention center, we have the Willamette River, where you can go for a walk or take a run, either before or after your day at the conference. Most of you will come into Portland International Airport. It's a very nice, award-winning airport. We also have Powell's City of Books nearby. It's the world's largest bookstore. Plenty of great food here in Portland, really active food scene. A little bit to our west, we have the Oregon coast. You could take a day trip out there, either before or after the conference. And a little bit to the east, we have the Columbia River Gorge. Plenty to eat and enjoy out there. That was McMenamin's Edgefield. This is the Multnomah Falls area. As you can see, obviously very, very beautiful. Back in Portland, we've got more parks in Portland than I believe any other city in the United States. So plenty to do and enjoy here. What I want to focus on right now is our conversation on Friday, which is going to focus on mitochondrial medicine. This will be introduced by myself, Dr. Alex Vasquez, in the morning, followed by Dr. Matt Hershey, Dr. Michael Gonzalez, and Dr. Garth Nicholson. We're dedicating an entire day to this topic because it's very, very important, as you can see from some of the articles that we'll review in this brief presentation. And so I certainly would encourage you to join us for this day. I think it's going to be very fun and very exciting. And so let's talk about why that is. Dr. Michael Gonzalez has published many articles on nutrition and metabolism in general. Those are certainly his major areas of interest. And one of his most recent papers, just published last year, called The Bioenergetic Theory of Carcinogenesis, connecting mitochondrial dysfunction with malignancy progression. This is one paper published by Matt Hershey late last year, came out. It was kind of pre-published in December and then officially published in January of 2013. Really nice article. Really helps us understand a lot about some of the effects of fasting and in particular the molecule beta-hydroxybutyrate, as you can see here. Garth Nicholson has published a lot of papers on chronic fatigue syndrome, Gulf War illness, and the role that chronic infections have in inducing and perpetuating mitochondrial dysfunction. You can see one of his papers here. Dr. Nicholson's published about 600 scientific publications, so we certainly have a lot to choose from when we review some of his work. This one obviously on the prevalence of mycoplasmal infections in patients with chronic fatigue syndrome and Gulf War illness. Another article by Dr. Nicholson here on metabolic syndrome and mitochondrial function and dysfunction, which I'll also talk about more throughout this presentation. So let's talk about mitochondria. We all learn about these in high school biology and college biology and then in medical school. And mostly what we learn is that mitochondria produce ATP. We also know that they house the Krebs cycle and the electron transport chain. And for many of us in medical school, we had to learn every step, every metabolite, and every enzyme of that step. I'll show you a graph of that later. I'm sure you've all seen it before. So let's talk about why is that relevant? I mean, if people are alive, doesn't that mean that they're making ATP and that their mitochondria, therefore, by definition, must be working adequately? The answer to that question, of course, is no. So let's talk about what can go wrong with mitochondria and what would be the clinical relevance of that. So here again is your mitochondria. You can obviously look at the structures. We've got the outer mitochondrial membrane, the inner mitochondrial membrane that houses the electron transport chain. And nutrients, of course, are very important for those biochemical reactions. Some nutrients, such as DHA, the fatty acid found in fish oil, actually anchors some of the enzymes to the inner mitochondrial membrane. So the nutrient in the form of a fatty acid is actually necessary for proper structure and function of the Krebs cycle and the electron transport chain. So when we talk about using nutrients, we're not simply using them from a biochemical perspective per se. They actually engage in the structural makeup of the mitochondria itself. So not just the biochemistry, but actually the structure. So let's talk about some things that can go wrong and lead to what we call mitochondrial dysfunction. Well, obviously, if people need nutrients, if the mitochondria need nutrients in order to function properly and engage in those biochemical reactions, nutritional deficiencies would be expected to impair mitochondrial function and therefore obviously lead to what we call mitochondrial dysfunction. So, nutritional deficiencies are quite common in the general population. They usually occur in groups. Nutritional deficiencies rarely occur in isolation. So, nutritional deficiencies can contribute to mitochondrial dysfunction. I think that should be pretty obvious. Mitochondria are very nutrient dependent, many biochemical reactions occurring, a lot of free radical production. So, if sufficient nutrients aren't available to engage either as cofactors or coenzymes or as antioxidants, that's going to impair mitochondrial function and lead ultimately to what we call generally mitochondrial dysfunction. Well, what about carbohydrate excess and especially high fructose corn syrup? That can also contribute to mitochondrial dysfunction. So can chronic infections and chronic dysbiosis. That can also lead to mitochondrial dysfunction. The evidence is very clear on that. And as I showed you just a second ago, Dr. Nicholson's focused a lot of his work on that topic. We all know about xenobiotics and chemical pollution that we're all exposed to. Many of these chemicals also impair mitochondrial function. A really great example of that in the literature is the association between herbicide and pesticide exposure and the development of Parkinson's disease. But some of these toxins are actually produced endogenously, like hydrogen sulfide and D-lactic acid. We get sulfite from either endogenous production or from the consumption of certain foods. And of course, as I mentioned before, we're all exposed to herbicides and pesticides, many of which are mitochondrial poisons. Oxidative stress itself can also impair mitochondrial function. So those are all things that can lead to mitochondrial dysfunction. Mitochondrial dysfunction then increases production of oxidative stress. Oxidative stress then causes more mitochondrial dysfunction. So you can easily see there that people can get trapped in a vicious cycle of oxidative stress leading to mitochondrial dysfunction, leading to more oxidative stress, leading to either the exacerbation or perpetuation of mitochondrial dysfunction. As more oxidants are produced, that leads to antioxidant vitamin depletion, and then that also leads to depletion of the other roles that those nutrients would have. For example, certain forms of vitamin E are necessary as antioxidants, but they also modulate immune function. Well, if those nutrients, and in this case vitamin E, if vitamin E gets consumed by free radicals caused by mitochondrial dysfunction, then that vitamin E is no longer available to modulate immune function and have its anti-inflammatory effect. So this is one way, among several ways, that mitochondrial dysfunction can then lead to secondary effects such as chronic inflammation and immune imbalance at the very least. Well, when we have increased oxidative stress and we have antioxidant vitamin and nutrient depletion, therefore, then those nutritional deficiencies can also contribute to the perpetuation of mitochondrial dysfunction, as you would expect. So as the mitochondria become more dysfunctional and produce more free radicals, they consume antioxidant nutrients at a faster rate. Well, that only makes the mitochondrial dysfunction worse. Oxidative stress also contributes to activation of NF-kappa B and what we call the inflammasome. That leads to more inflammation, which, as you would expect, leads to more oxidative stress. So you can see that oxidative stress and mitochondrial dysfunction in particular can originate from several different causes, and that it can also become a vicious cycle perpetuating itself. So let's look at some more important concepts. This was a really great article published in the August issue 2011 in Science. Let's read a few sentences from this article together. Mitochondria participate in the detection of infectious microorganisms and cellular damage to activate the innate immune responses. Wow, that's pretty amazing. Most of us didn't learn that in medical school. We only think of mitochondria in terms of ATP production. A few of us think of mitochondria associated with this enigmatic phenomenon called apoptosis. But let's look at this sentence one more time. Mitochondria participate in the detection of infectious microorganisms and cellular damage. Those things activate what I referred to before as the inflammasome to activate innate immune responses. it's likely that the accumulation of damaged mitochondria is an important cause, in this case, of chronic inflammation. The removal of mitochondria that have a rather low threshold for permeabilization is called mitochondrial purging. And I'll show you a little bit more about that. Obviously, we'll talk about that more during the conference, but I'll show you one way to induce mitochondrial purging when we go through the material in this presentation. So why does any of this matter? For those of us who are seeing patients, why do we care what's going on with their mitochondria? Well, obviously, it's because mitochondrial dysfunction can lead to certain diseases. Let's talk about those. Chronic inflammation. Mitochondrial dysfunction contributes to cell senescence, that's cellular aging, chronic inflammation, and what we call inflammaging, which is the connection between inflammation and what Jeffrey Bland has referred to as rapid biological aging. Patients with allergy often have mitochondrial dysfunction, and mitochondrial dysfunction promotes an inflammatory climate, and that can contribute to conditions like asthma. What makes the situation worse is that some drugs, like prednisone, for example, which is used to treat asthma, actually make mitochondrial dysfunction worse. Mitochondrial dysfunction is very common among patients who have autoimmune conditions like lupus and rheumatoid arthritis, and especially metabolic syndrome, diabetes type 2, and hypertension. Mitochondria, most people don't know this, but mitochondria are required for insulin secretion and insulin reception. So, if a patient had mitochondrial dysfunction, we might expect them to have difficulty either properly secreting insulin or properly receiving that insulin in the peripheral tissues. And that is certainly the case. I'll talk a little bit more about that in this presentation. Heart failure kind of makes sense. The heart is obviously very dependent on ATP production. It's dependent, therefore, by definition, on proper mitochondrial function. If those mitochondria aren't working very well, then we would expect heart failure to be a potential result. Fibromyalgia, likewise. Histologic and biochemical evidence of mitochondrial dysfunction, mitochondrial failure, and mitophagy. We'll talk a little bit more about fibromyalgia in this presentation. Migraine headaches, I'll show you a diagram that I made showing biochemical evidence of mitochondrial impairment, and of course, neurodegenerative conditions like Parkinson's and Alzheimer's. I would like to think that everybody pretty much knows this by now. It's been published for a long time. In fact, I've published some review articles on this many years ago, back in 2004, I believe, 2005. So I think that this kind of information should be pretty well known. And for those of you who don't know it, this is certainly a chance to learn about it. Anytime you deal with a patient who's got Parkinson's or Alzheimer's and several other neurodegenerative disorders, you've got to think of what's going on with their mitochondria. So that's my first slide, connecting mitochondrial dysfunction with disease. Let's see if there's a little more information for us to look at. So these are just quotes from the literature. Here is Journal of Immunology 2009. Mitochondrial dysfunction increases allergic airway inflammation. So obviously that's reactive airway disease or asthma. Mitochondrial dysfunction increases the inflammatory responsiveness of to cytokines in normal human chondrocytes. That's published by the American College of Rheumatology last year in 2012 in their journal Arthritis and Rheumatism. Likewise, mitochondrial dysfunction activates cyclooxygenase 2 expression in cultured normal human chondrocytes. So we all know that there's a whole class of drugs out there called COX-2 inhibitors, cyclooxygenase 2 inhibitors. So why would we need to inhibit cyclooxygenase 2? Perhaps it's due to mitochondrial dysfunction. Therefore, perhaps the more logical therapeutic intervention would be to address mitochondrial dysfunction rather than treating the downstream effect of it, which is increased COX-2 expression. Anytime during the video, you, of course, can hit the pause button and slow down and actually read this word for word if you want. Mitochondrial dysfunction, according to this quote from Arthritis and Rheumatism, sensitizes cells to amplifying the inflammatory response induced by cytokines. This is an article that we looked at a few moments ago, suggesting that specific treatment of mitochondrial dysfunction in intensive care unit patients may significantly improve the clinical outcome. Finally, Journal of Neuroinflammation, effects of mitochondrial dysfunction on the immunological properties of microglia or microglia. So we'll talk about that too. On Thursday, we have some really fascinating presentations on brain nutrition and brain health, and we'll definitely talk about not only microglial activation, but also astrocyte activation, which is, again, here relevant to our conversation on mitochondrial dysfunction. So I told you we'd talk a little bit more about mitochondrial dysfunction and fibromyalgia. Very fascinating topic. I've written 2 books on this topic, actually. These patients often have nutrient deficiencies. One of the deficiencies that they have, in addition to several others, is reduced levels of CoQ10. That's because their mitochondria are kind of over-consuming the CoQ10 because they're in a chronic state of dysfunction. Increasing free radical generation that consumes the coenzyme Q10, then the CoQ10 can't be around for its other functions, such as contributing to the function of the electron transport chain, as I'll show you in just a moment. As I mentioned before, mitochondrial dysfunction also contributes to Alzheimer's disease. And another topic that we see very often in our clinical practices is the topic of diabetes, hypertension, and metabolic syndrome. Let's look at the connection between those conditions and mitochondrial dysfunction. Really fascinating work here, published back in 2008. Let's look at the quote here. Insulin resistance emanating from mitochondrial dysfunction may contribute to metabolic and cardiovascular abnormalities and subsequent increases in cardiovascular disease. Therefore, interventions that improve mitochondrial function also improve insulin resistance. Well, here again, we have a whole class of drugs designed to improve insulin resistance, but if that insulin resistance is caused, at least in part, by mitochondrial dysfunction, the wiser clinical strategy, of course, is always to treat as far upstream as we can. And in this case, we should be focusing on the component of mitochondrial dysfunction in diabetes, hypertension, and metabolic syndrome. Let's look at mitochondrial dysfunction in migraine headaches. So here's the Krebs cycle, and you can see at the bottom the electron transport chain. The blocks that I've highlighted in red are the defects seen in patients who have migraine headaches. So anytime you deal with a patient who's got any type of chronic primary headache syndrome, such as migraine headaches, you are definitely dealing with mitochondrial dysfunction. In fact, an article that I looked at recently stated that the severity of the clinical phenotype of primary headache syndromes, again, such as migraine headaches, is directly proportional to the biochemical impairment of the mitochondria. So the worse the headache syndrome, such as migraine, the worse the headaches, the worse is the mitochondrial dysfunction. And obviously we have several interventions we can talk about to improve mitochondrial function and therefore alleviate the migraine headaches. Some new research came out earlier this year, in June actually, so it was just last month, June of 2013, changing the way that we view the electron transport chain. If you look at the previous diagram, which I suppose I could go back to, let's look at the bottom of the page here and just look at the electron transport chain. Electron transport chain typically has 5 of what we call complexes, or 5 steps: complex 1, complex 2, complex 3, complex 4, and then ATP synthase, often referred to as complex 5. So typically we've looked at those in terms of it being a rather solid and linear model. What came out last month in Science was a new article, obviously kind of changing the way that we see that. Instead of seeing the electron transport chain as a linear model, now we appreciate it as existing in the form of supercomplexes. And those supercomplexes, the structure and components of the supercomplexes are actually changed through nutrient intake. So supercomplex assembly is variable dependent upon the diet. Very fascinating work. We'll talk about that during the conference. Let's talk a little bit more about mitochondrial dysfunction. So as I said before, mitochondrial dysfunction contributes to chronic inflammation, diabetes, hypertension, asthma, Parkinson's and Alzheimer's, and of course fibromyalgia. We have at least 26 interventions to improve mitochondrial function, summarized in my book that I hope to get out at some point, called Mitochondrial Nutrition. I had hoped to publish this in 2012. Didn't happen. I've got a lot of the work ready, but maybe later this year it'll be ready, but you'll get that information during the conference. So let's talk about some treatments for mitochondrial dysfunction. We have at least 26 interventions to improve mitochondrial function. Again, these eventually will be summarized in my book, but you'll get them in the conference ahead of time. One of the treatments we can use for mitochondrial dysfunction is actually fasting. So when the body undergoes this fasting state, it purges or deletes dysfunctional mitochondria. So one of the strategies for improving global mitochondrial function is to delete the mitochondria that are not functioning properly. And one of the ways we can induce that is through fasting. really fascinating work there. Obviously fasting has been used in many different cultures for many different centuries and millennia for that matter for various, social and religious and spiritual purposes, and obviously for health purposes as well, usually in the treatment of chronic infections or chronic rheumatic disease. Well, how would fasting help a patient who has chronic inflammatory disease? One of the ways it helps, and it does work through several different mechanisms, not simply weight reduction and not simply being the ultimate low allergy diet, but also through promoting the mitochondrial process of what we call mitochondrial purging of dysfunctional mitochondria, also known as autophagy or mitophagy. So when people fast, they actually start to delete their dysfunctional mitochondria so that a new population can spring forth of healthier mitochondria. Very fascinating to understand the mechanism of fasting from that perspective. So there are lots of other treatments we can use for improving mitochondrial function because, not all patients are candidates for fasting. Some patients have chronic, herpes infections, for example, and so we can also improve their mitochondrial function by suppressing their viral replication. So, worldwide, about 50% of people have herpes type 1, about 20% of people have herpes type 2. So you're looking at about 70% of the human population has some type of herpes type 1 or type 2 infection. Obviously, there are other herpes viruses, which can also impair mitochondrial function. So another approach to improving mitochondrial function is to implement some type of safe, long-term strategy to suppress herpes virus replication so that the herpes virus isn't actually destroying mitochondrial DNA, because obviously that would lead to loss of function. So one of the strategies we can use to improve mitochondrial function in our patients is to suppress viral replication. So we have lots of strategies we can use. We'll talk about those during the conference. I think you'll find that quite fascinating. I certainly find it fascinating, which is part of the reason why we're talking about it right now. So I hope you'll be able to join us for our nutrition conference here in Portland, Oregon. Again, that's going to be September 25th through the 29th. We've got a lot of great speakers. These are all detailed on our website and in the brochure that you can download from the website. You can obviously get a lot of information from our Facebook page as well. Our Facebook page has become very popular in just the few months that we've had it up. And one of the things I post up there periodically is information about the venue. For example, we just got another hotel discount block, this time at the Hilton Doubletree Hotel, which is just a few blocks away from the convention center. In fact, from this room in particular, you can actually see the convention center from the room. It's only about 3 blocks away, as you can see here on the map. So that's another option. You can get the details from our Facebook page. Another thing that I've added recently to our Facebook page is kind of an ongoing quiz or challenge, you might say. So here's the case that I put up recently. Actually, I just put this one up last night. So here's our weekend case study, in case you want to follow the discussion thread on our Facebook page. Your new patient is a middle-aged male who presents with peripheral polyarthralgia, fatigue, and an evident case of definite psoriasis following administration of amoxicillin for a dental procedure. What is your initial assessment and treatment plan? So this, I think, will be a pretty fascinating case, especially for those of you who are new to integrative care or what we generally refer to as functional medicine. I think you'll find this case workup to be very interesting. I'll post some more replies soon, and eventually I'll post a very quick review video as well. I think you'll see that my evaluation of this patient was pretty interesting and very detailed, but also very practical and very affordable. For those of us, for example, who don't take insurance in our clinical practices, we always have to be mindful of being of trying to be very effective, of course, but at the same time, not spending a lot of money on unnecessary lab tests. So we have to become very wise in our selection of laboratory tests and our therapeutic interventions. So we look forward to seeing you at our international conference on human nutrition and functional medicine. We have a lot of information on the website and on our Facebook page and our growing list of videos as well. So look forward to seeing you there, and I will update everyone with more information very soon. Thank you much.